Hello, welcome to another A-level lesson from the conscientious biologist, Ben Gallagher. This one looks at mitosis and the cell cycle. You don't strictly have to have watched any other videos before this if you just stumbled onto this and want to revise it as a complete thing, though it is useful to have a solid knowledge of DNA structure, DNA biochemistry, and a good overview of cell biology and cell structures. As always, please do subscribe to my YouTube channel if you haven't done so already, and do remember you can find me on both Facebook and Instagram for lots of additional biological information. Thank you. So first off, let's ask the obvious question and make sure you understand what is mitosis. Well, it's normal cell division that you have in eukaryotic cells. There's different types of cell division in other cells. Binary fission is the one that bacteria use. So this is normal cell division in eukaryotic cells. That's plant, animal, protoctist, and fungi. Done in slightly different ways in some of those other kingdoms, but it's much the same. Now, it's the process by which cells make copies of themselves. So if you start with one cell, one's going to divide into two, each of those into two, so you get 4 into 8, 16, 32, 64, 128, and so on and so on. It's the way that cells increase in number. So if we're thinking about why they do that, well, it's cell cloning, and the reason for that cell cloning is for growth. As you get bigger, you need to be made of more cells, but also for repair. If you damage some cells, the ones around them have to do mitosis a bit faster to replace that damaged tissue. So mitosis, it's cellular cloning in eukaryotes for growth and repair. That's the obvious. That's kind of what you're told even in year 7. We cover details like that. Now we're going to fit that into the cell cycle as a whole. I'm going to represent the cell cycle by this circle, but that circle, try and think of it like a clock face where I'm going to go through periods of time. Okay. Now the first bit of the cell cycle I'm going to mark off in pale pink here, far and away the largest section, is interphase. Now interphase is just the cell's life. That's where it's just doing the stuff it needs to be a cell. So it's where it's performing all its metabolic reactions. Of course, probably the most important one of those is the protein synthesis. So it can make all the proteins it needs, so those proteins can carry out all those metabolic functions, things like respiration or photosynthesis if it's a plant cell. So really, really important. That's when it's doing what it needs to do. Of course, during interphase though, it also needs to double in size, because if you've got a cell that big like that, if it's going to divide in two, if it just divided in two like that, you get tiny little cells. And then when they divide, they get smaller and smaller until they disappear. You need a cell to double in size so it can pinch, so each new cell is the same size as the original, these circles I'm making with my fingers. Okay, so it's got to double in size. Now, really importantly, from the last video that hopefully you watched, it's got to copy its DNA by semi-conservative replication. That's so each of those new cells can have a set of DNA of its own, not just 50% of the DNA, which would then get half to 25 and then down to 12.5 and so on. They need 100% of the DNA. So the 100% that was originally in the cell needs to turn into 200%, so each one can have a copy. That was what we talked about in the last video of going single chromosomes to double chromosomes. But when does it do that? Well, it does it at a distinct point of the cell's life that we call S phase. I've just put here and marked with an S there. Now, pretty much everything else in the cell needs to shut down while the DNA is replicating, because the DNA's instructions for how to make the proteins that do everything. If the DNA is busy dividing, it can't be read, so a lot of the metabolism shuts down. So we can split interphase now into two sections, G1 and G2. Now the G you can think of as growth 1 and growth 2, but it's kind of like the interphase stage 1, which is G1. Then you've got to replicate your DNA. Then you can go back to what you were doing, back to interphase. So you can kind of break interphase down into G1, S, and G2. Now, the bit we're talking about in this video is the other pink corner over there. So everything circled in that black, that's interphase, covering G1, S, and G2. We're interested in that pink corner now, which I'm going to divide up into 4 sections. That's when mitosis happens, and mitosis is split into prophase, metaphase, anaphase, and telophase. Collectively, those make up mitosis and cell division. So very simply, you could break a cell's life down to interphase and mitosis. That's pretty much it. Okay, let's start to look in detail at what's going on in mitosis now. Right, so that we can start mitosis, let's think about the very start of mitosis would be the end of interphase. So let's just make sure you're happy with what the end of interphase would look like. So if this was the very start of interphase, your brand new cell, we know that during interphase that's got to double in size. I said to you before that it's got to double in size, so when it divides in two, those new cells are the same size as the original cells, so they can keep doing mitosis. But as well as that, it has to change these single chromosomes in its nucleus into double chromosomes, because a double chromosome is effectively just two copies of the single chromosome, but we call them sister chromatids, remember? That's so that each one of those can end up being in one of the new cells that's created. So you've got to have this semi-conservative replication of DNA to put double chromosomes into the nucleus of a cell at the end of interphase. So remember, that first one is G1. This one closest to us is now G2. So G2 really is our very start point for mitosis because it's the end of interphase. Now, just a couple of key points to make sure you're happy with. By the end of interphase, cells should be twice as big and have double the quantity of DNA so that they're ready to divide, so we can start mitosis. And they did all of that stuff whilst also performing everything else that they're supposed to be doing, all their normal metabolism. Now, really, really vital point. My diagram's slightly misleading because chromosomes would be diffuse during interphase, so they wouldn't actually be visible. So if you were looking at a cell in G1 and G2, you wouldn't actually see the chromosomes like I've got here. I've only made them visible, so it's easier for you guys to see what's going on. But they'd be diffuse and spread out, because by being diffuse and spread out, the DNA can actually be seen and be read, so it can be expressed into proteins. Okay, so we'll include the chromosomes on here, but remember, you wouldn't see them if you were looking through a microscope. So really making a start on mitosis now. We've come out of that G2, we're going into the first phase, which you'll remember was prophase. Now, prophase is about preparation. So for each phase, I've given you a word next to the title that will help you remember what's going on in that stage. And I've coloured the P blue, same as prophase, prophase P, preparation. Okay, because what's going on there? If we've got our cell from G2 from the last diagram, to achieve prophase, or the preparatory events of prophase, there's three of them, three key events. The first one of those is that the diffuse chromosome condenses into visible double chromosomes. So on the last slide, I said that these are a bit misleading. So it would only be now that when they condense, they become visible like you can see on the diagram there. Okay. Event 2 is that the nuclear envelope, remember that from the cell ultrastructure, the nuclear envelope will break apart. That's to release the chromosomes into the cytoplasm. So they're now floating around. Now you've got to release them, because if you're going to separate them and break them into two different cells, you can't do that if they're contained in a nucleus. So you have to break down that nuclear envelope, that double layer of membrane, to release them. But of course they are a bit vulnerable now because they haven't got the protection of the nucleus. But they are kind of protected by being so tightly bunched up and coiled that you don't need to worry about it too much. Now for the third event, we need to remember something specific from the cell ultrastructure lesson. And that's these little things, okay? Centrioles. Centrioles we talked about in ultrastructure lesson. This was the slide from the ultrastructure lesson. Centrioles are made of microtubules. In animal cells, you have centrioles that look like this. If you can't remember it, go back and watch the ultrastructure lesson. But the centrioles sit just outside the nucleus. I'll put them over on the cell in G2. They're at right angles to each other, and you've got two of them. Now what happens as the third event is those centrioles start to migrate to opposite ends of the cell. So what you've actually got here now on this picture, this is representing prophase. The chromosomes are now visible because they've condensed. They're floating around in the cell because the nuclear envelope is broken down to release them. And the centrioles have started to migrate to opposite ends. All three of those are just getting ready for the cell to divide that DNA up. Take a screenshot of this one because this is the first stage done. Okay, so prophase was first, so moving on to metaphase now. I'm going to pull up the prophase from the last diagram so we can see where we were to see what we progressed to. But I'm going to introduce a phase that you don't strictly need to know the name of, but it just makes sense to explain it. It's always annoyed me for years teaching this, that when you study mitosis, it's like you've got a flick book and you're flicking through the pictures to try and turn it into motion, but you've only got every kind of 10th page, so you can't really tell what's gone in between. So you've got this snapshot of Prophase that looks like that. But remember, in the last slide, I animated it to get to that. But Prophase itself, you don't see the events that took you from G2 to Prophase. So I'm going to try and fill in some of the in-betweens now by talking about an in-between phase to get us to metaphase. So we can see where we were on that last one. What happens though is those centrioles keep going to get to opposite ends. Once they're at opposite ends, the centrioles, which remember are made of microtubules, they fire out things called spindle fibers. It's just that the microtubules extend. Those spindle fibers shoot out into the cell from both sides, and they'll hit the double chromosomes. They attach to each double chromosome at the centromere, so that, now we can go to metaphase, they're pulled to the middle of the cell. So the centrioles have to get to the outside, fire out their spindle fibers to grab the chromosomes, then pull them to the middle. Okay. Now that in-between phase, because it's in between prophase and metaphase, is generally referred to as prometaphase, okay, because it's in between. But it's really important that you understand how we got to this metaphase here from the prophase over there. And it's by going through there that these spindle fibers pulled them into position. Now for metaphase, the little code key I've given you there is move to the middle. So you've got two M's to go with metaphase. If you see a picture like this one, it's metaphase. Okay, let's look at the next one. So after metaphase, we have anaphase. So let's pull up the last one, same as we've done on the previous slide to see where we were. So metaphase looked like that. All that happens in anaphase is that the spindle fibers retract back into the centriole. So it's like they're pulled in. Okay, it's like the centrioles are pulling them back in. Now, if both centrioles pull from both sides, the double chromosome is going to get broken at the centromere. So what happens is the double chromosome, each double chromosome, try and use that word each to avoid ambiguity. Each double chromosome is broken at the centromere to separate the identical sister chromatids back now into single chromosomes. So you've now got, technically in this cell, you've got 92 chromosomes because they're single. You started off in G1 with 46 single. You copied them up by semi-conservative replication to 46 double chromosomes. Now at anaphase, because you've broken each of those double chromosomes, now for a brief period, you've got 92 single chromosomes in here. Okay, those identical sets, because remember the two sister chromatids identical to each other because of semi-conservative replication, they're pulled to opposite ends of the cell. This is anaphase, and the little clue that I give you for that is it's when they pull apart. Think of the a for apart goes with anaphase. Okay, let's look at the final stage then of telophase. Okay, so again, we'll pull up the previous one over there. So we've got the last anaphase one that we had. Now let's just shuffle that along to slightly further along. You can see the chromosomes have just been pulled a little bit further towards the poles. Now, the first thing that's going to happen in anaphase is that the spindle fibers break down. So you'll see I've made them disappear from this closest one. We don't need them anymore because the chromosomes are where they need to be. Second event, new nuclei, so a new nuclear envelope will form around each of those two sets. So we now in this cell have two nuclei. Each nucleus contains 46 single chromosomes, but the cell as a whole still contains 92 single chromosomes. That's the total of both the nuclei. Okay. Next event is that the cell pinches in the middle by cytokinesis. This is, if I do it up the way that the page is, that's where it is that size. It pinches in the middle. So that's the picture that you're looking at now. And it's going to eventually pinch all the way through. But that pinching that you can see all the way around, that's called a cleavage furrow. Now, when the cleavage furrow has pinched in enough through this process of cytokinesis, which is really important, it's going to pinch the cells in two. You're going to get two new cells. Now, that whole thing there, that's telophase. Okay, the getting rid of the spindle, the formation of the nuclei, the pinching of the cell, right the way through to the final cytokinesis that splits them in two. So I put telophase here, and for telophase, easy one, telophase is when it divides in two. T for two, T for telophase. Okay, So you've got to remember all of the events that happen all the way through. Now, to help you do that, I'm going to give you a resource on the next slide. So we're going to put the whole thing together on this slide, one step at a time, but I'm going to leave you all this white space so that you can screenshot this, print it out, and then you can write your own explanations in. Print it out 10 times and at random points leading up to your exams. Just keep practicing. Just get a blank copy of it and go, right, and describe, you know, what took me to G2 during interphase. Now we're on mitosis. What are the 3 events of prophase? What happens at metaphase, and blah, blah, blah. Okay, but here we go. We've got G1 that led us into mitosis. G2 is still part of interphase. Remember, G2 is not part of mitosis. But for prophase, those 3 key events of the nucleus disintegrating, chromosomes coiling up, centrioles migrating. Then I'm going to include the pro-metaphase on mine, just to show how you got to metaphase, for the chromosomes aligning in the center, pulled there by the spindle fibers. Now, anaphase, those spindle fibers continue to pull, but because they're now right in the center, the only thing they can do is pull them apart, breaking them into single chromosomes, so that you can enter telophase to repackage those chromosomes and divide them into 2 new cells. So screenshot this, print it out, annotate it. That's yours to use to help you revise. Now, as well as knowing the process, in the exam, they quite often expect you to be able to identify cells at different stages. So what I want you to do first is pause the video for a minute so that you've got time to look at this, and I want you to try and identify as many stages of as many cells as you can. So pause the video now and do that. I'm going to hope that you did pause it and look at them, so I'm going to carry on now. And we're going to look at some together. So if I just pick out this one over here in the blue square, I picked that one at random. Loads of them look like that because that's a cell at interphase. Can't tell really what stage of interphase other than by looking at its size. It looks pretty big to me, so I think that's probably in G2. Now, the reason why I know that's in interphase is because I can't see any definite chromosomes in there. The nucleus is there. The nucleus is the darker purple blob. The paler blue is the cytoplasm behind it, and you can just about see, these are plant cells, so you can just about see cell walls on there, that the even darker blue bits around the edges. But what we're looking at on there is a nucleus in purple with no distinct chromosomes. So that's got to be interphase. If I mark this one now more in the middle, have a look at that one, just decide for yourself what you think that one is. Okay, I think that that one's in early prophase. The reason why I think it's early prophase is because you can still you can see chromosomes now, so you know they've started to condense. But they all still, to me, look like they're in the shape of a nucleus. So the nuclear envelope clearly hasn't really broken down yet. So we're at an early stage of prophase. If I mark another one over there, that one and that one, so I'm marking 2 up there, they're both in virtually identical phases. I would say both of those are in metaphase. Hard to tell whether it's early or late metaphase because you don't know how spread out the chromosomes are going to be. I would say both of those are at pretty early metaphase, because the chromosomes are still sort of pointing out into the cell. They're probably on their way to being pulled in. However, they might be in early anaphase, and maybe they're so pointing out because they're already starting to be pulled apart. But certainly that one up there, that one is definitely early anaphase, because you can see the chromosomes have been separated from each other and being pulled to opposite ends now. This one, if I mark over there, you can see is definitely quite late anaphase. The chromosomes are much further apart than they were in that early anaphase one up there. And if I mark this final one over there, that one looks to me like it's in telophase. You can see the chromosomes still, you can see they're at opposite ends, you can see they look repackaged into nuclei, but the cell hasn't quite divided in 2. So I would say that that one is definitely in telophase, probably relatively early in telophase. Now, screenshot this, print it out, add it to your notes, whatever you need to do, because this is almost always asked in the exam for you to comment on the stage that something's in. They'd probably only give you 1 cell, not a whole cluster like this, but they'd probably give you 1, could be a picture of any of these, and say, what stage do you think this is in, give your reasons. And you'd need to pick your stage and say, because I can see X, Y, and Z events are going on. Okay, it's a really important skill to develop. Right, really quick final slide, and this is just to make sure the numbers make sense in your head. What I've got here, I'm going to put it all together just very simply as a summary. So we start with one cell, it gets bigger, it doubles its DNA. That's going to then divide into two. That's the process of mitosis. If we're putting this in humans, in human context with the numbers, the cells will be genetically identical to each other, but in terms of numbers, that original one had 46 single chromosomes. That's what we have as humans. The middle one there has got 46 double chromosomes because the cell underwent semi-conservative replication to double its quantity of DNA, but the number of chromosomes is the same because you went from singles to doubles. Through mitosis, you separate the doubles into their two copies, split them into new cells, so you end up with 46 single chromosomes in each. This is just a very quick summary so that you've got the numbers in place. And that's it for this one. Mitosis, big process, almost always comes up in the exam. They'll either ask you to identify a cell and tell you why. They might sometimes ask you to describe the events of mitosis. That could be a really big mark question, or they might, say describe the events that occur in prophase or whatever. But you've got to be able to describe it with lots and lots of detail. Think back to those ones that I did before. I put the keywords in purple. That's what you must be able to do. Okay. From here, go to cell differentiation, please. That's the most logical video to watch next. Give this video a like if you find it useful. Please do subscribe to my YouTube channel and do check out my content on both Facebook and Instagram accounts. Thank you.